Hello everyone. So this is the last session of this the whole program. This is going to be we plan to have the interactive session. So just to go through overall um, from the day 1 to day 5 from morning session that what we discussed. So this is going to be more of interactive just to go over what we learned all these days and also kind of all the models that we developed, and just to have the interactive session after I go through some of these models that we discussed all these days. Okay, so overall the in the model that we discussed about the in vitro models, the cell line model, 3D culture, some, and organoids. I just touched on the organoids. When if you want to do the organoid culture, how you do it and whether you want to do it, and if you want to do it, how to do it there's a procedure. So we can actually, there are a lot of beautiful papers published on methodologies. So you can just say organoids and methods. There are lot of beautiful papers. So in our laboratory also we follow those methodologies from the pioneer who worked in pancreatic cancer, and then we follow their paper, methodological paper. And the same way, if you have a different cancer, different method, you can actually follow the same. And more over that, there are a lot of discussion forums online that you can actually, if you have any doubt and questions all over the world, people discuss about a lot of problems and issues. So that's what I think during when we were learning. So we discuss into the forums, but nowadays we meet them in the conferences and we go to the posters and presentations. Organizers who are actually working on, they're all pioneers, so they might have learned a lot. So we go and discuss during the poster session and also during the presentations. So what I suggest, you go to these conferences if you are interested in some particular aspects of your studies, and go to those posters where they've used it already, search for those keywords or go through those posters and talk to them and discuss with them. And they are the one actually real biologists like working on those technologies. So 3D culture also, it may feel like simple, but it may not be simple. Okay, there are a lot of errors and things that when we start working. So go to those posters and go to conferences. That's why these meetings, for. So to meet other investigators who are actually working in that particular field and interact with them, right? So in vivo model the same way. We talked about xenografts. We talked about syngenic models, GEMS, patient-derived xenografts. Any of these questions or anything issues, or if anyone wants to work on, there are a lot of these conferences you should go to, symposias, or someone is presenting something on related to somewhere else. See whether you can have a Zoom call or online web media or related discussions. There are a lot of universities. They do have a open format, like anybody from anywhere in the world, they actually access those webinars, and you can actually listen to them, what they are doing and conducting experiments. So don't have the fear. So you don't to click those Zoom links and webinars. So you can actually, anybody can go to those webinars, attend and learn. Okay, if you are interested, there are a lot of them. You need to start searching. Searching itself is a huge thing. Like you learn a lot. For example, if you are interested in cancer biology, there is ASCR, right, American Association for Cancer Research. And the same way here in Indian Association for Cancer Research, they may be conducting the webinars, and ASCR conducts a lot of webinars, and also they conduct, for example, a Cancer Discovery Journal, right? They conduct the webinar. Anybody can access those webinars. Okay, means that journal club. Someone from that paper that who published, actually they talk about their paper and technology and how why did they do those experiments, exactly like how I was actually telling you about my story and my papers. The same way they talk about those papers, and you need to listen to them, understand what they're doing, if they are, if you're interested in those studies. We did not touch much about bioinformatics, but bioinformatics is a tool. I did talk about, our program, other programs, and programming, and single cell sequencing, RNA sequencing, data analysis, and when you run the flow and flow analysis and all this analysis itself, I think you need experts. Okay. So that, and then in vitro model we touched even today morning about the 2D culture, high throughput screening, and a 3D culture and organoids. We did talk about a little bit here and there, and the reproducibility issues and the lack of microenvironment. We talked about all these contents, like why microenvironment is important, when we should use these microenvironment related studies, when we should not. Okay, just stick to a xenograft model and keep going. The important is what challenges have you encountered within when you are conducting in vitro experiments and models, and how have you been overcome them. That's the question that you have to think. If you are not yet overcome the problems, then start thinking about whom to contact and get the information, right, or discuss with them. When we come to in vivo model, in vivo model, we talk about in vivo model because we want to bridge the lab to clinic. That's why we want in vivo model. Otherwise, we can do a lot of in vitro and biology studies, right? So we want to take that to the clinic. So that's why we want to bridge that gap between clinic to the lab, right? That's why we make the in vivo models. So we make different in vivo models, like for example, xenograft models, for a reason. Just for the tumor growth, drug testing, that's all. That's all. So if you are thinking about taking a xenograft and looking into some other marker, some other microenvironment, may not be the right model, right? So that's why you need to know what model to use. Okay, syngenic models. So these are immunocompetent system, ideal for the immunotherapy studies, right? So if you want to do the immunotherapy, these are the right models. The GEMS and PDXs are reflect the human tumor heterogeneity. So that's why they go for the GEMS and the PDXs, stromal interactions. So these are all microenvironment if you want to study. Those are the models that you want to, do. I shared my experience with the in vivo model selection and all the limitations that I went through during our work and in our laboratory. I've already discussed a lot of things that if you got the information, it's fine. If not, it's fine. So at least when the problem comes to you, then what you need to do, right? Reach out to someone who is expert in that particular area, particular model, and things like that, right? Okay, and then the competition biology, very important. Nowadays especially, you cannot just do alone. Science is going beyond. Every other day, there's a beautiful papers coming out in publications. So it's okay. If you are sleeping more, then believe me, that sleeping hours, the more like 2 hours extra, the when someone is publishing another paper. So think about it. And if you want to do research, there is a limitation. So how much you can enjoy, and also how much you have to spend time for each thing, and where you're wasting time, and where you can put those time in what for, things like that. You have to really judge yourself and be competitive. That's all I can. That's what I'm trying to tell you. So when it comes to bioinformatics, genomics, transcriptomics, proteomics, and you need to know those. If you want to do all those things, and you never know. Probably in the future, in another few years, probably you'll be one of the expert in transcriptomics studies or proteomics. Probably you'll be in industry and doing something like all this high throughput omics technology. You never know. So think logically. The models that you use, it doesn't matter what bioinformatics tool you're using it, but there's a logic behind each technology. I've already discussed, like you don't have to do genomics, proteomics, and transcriptomics. It all depends on the model that or the study that you're trying to do. There are a lot of virtual screening, a machine model predicting therapeutic response. There are a lot of models available. It's about, you don't have to think all of them, study all of them. Remember I told you? So you don't have to. You can learn on the way, but you just focus on something, take some problem, starts working, right? important is when it comes to a computational tool to understand what computation tool have been most impactful in your research. You need to find that, and also you look for someone expert in that particular aspects of computation tools that to use. For example, R. Why you want to use R? Okay, what for? If you do this analysis, what computation tool you need to use, and who is your friend in the bioinformatics department or somewhere else in the bioinformatics, or the computers, someone like in a statistical analysis, someone who knows actually statistical tools, things like that. So it's all about collaboration and thinking about friends and colleagues and around the world or around India. So therapeutic strategies, we talked about small molecule inhibitors, monoclonal antibody. We did not touch in detail monoclonal antibodies. So all you can talk about that if anybody is interested. It's a gene therapy. We can talk about targeted when it comes to targeted therapies. Immunotherapies, we talked about immunotherapy, checkpoint inhibitors like PD-1, anti-PD-1. There are other CAR T cell therapy, cancer vaccines, and there are a lot. You can, if you think about if you want to do the CAR T cell or the cancer vaccine, you can learn vaccine, how to do the vaccine, and go to that field and learn about it. It's all about your interest, though, end of the day. When it comes to chemotherapy, radiotherapy, these are all conventional therapies. They are still in use. They do in the hospital. That's all they do. Chemotherapy, radiotherapy, or surgery, and then chemotherapy, radiotherapy, adjuvant therapy and neoadjuvant therapy. They call in their terms in the clinic. So if anybody patients in the hospital, you can hear all those adjuvant therapy, neoadjuvant therapy, these kind of things. You as a cancer biologist, you need to know all those words. Important. The clinical words are important to understand what exactly is going on in the hospital, so that you have to mimic all those things in the laboratory. And when it comes to combination therapy, right, integrating multiple modalities. And also I talked about overcoming resistance. I started the talk with the overcoming resistance. The reason is, I'll tell you, I told you that that pancreatic cancer, right? So the same way that different cancer have different reasons to start with, and also when it comes to multiple modalities, integrating different technologies, studies, and models, and we talked about and touched all those things. So also you need to know that which therapeutic strategy has shown the most promise in your study is a key thing, right? So you need to know what strategy you should use in the therapeutic strategy you should use it during in your study. So like you need to find out your own way, and also the key aspects concepts are in the targeted therapy. Focusing on the molecular target. So for example, we talked about one particular kinase, one particular transcription factor, oncogene. So think about it. I talked about KRAS, I talked about P53. Think about why you want to target this. I gave you a lot of logics behind why I want to use KRAS model, why I want to use the P53. The logics behind. So you cannot just go ahead and start working on something like I want to start. There's a logic behind in each and everything. That's why this takes, you need to do a lot of literature survey. That is the reason. So, for example, we talk, there's a EGFR inhibitor. I touched, in our screening, the EGFR came up. PARP inhibitor people use, BRAF, inhibitors they have used. So these are all targets. So you need to know what target you want to touch and why and all those things. So there are. You also should be able to judge yourself how you want to address this resistance pathway. So in any cancer you take, okay, resistance mechanism is an important thing when using the targeted therapies, right? So you need to know when this tumor will be resistant to some therapy that you are doing, and how do you overcome. So I gave you a lot of examples in overcoming resistance in our models, and you think about your models and your studies in the future. Okay, the types of immunotherapy. There are many types of immunotherapy. We use one. I presented one anti-PD-1. There's a paper that we published, Cancer Gastroenterology. We used even CTLA4 in that. There is another checkpoint inhibitor. there are cancer vaccines, cytokine therapies. So we have done cytokine therapies too. So there is a reason why we do the cytokine therapy, and also which cytokine we use as a therapy. There are benefits, limitations in each one. If you come to immunotherapy doesn't work in the pancreatic cancer, just the immunotherapy. You have to add with the targeted therapy, chemotherapy, and other therapies. So there is a reason we can, like as you do research and establish papers and knowledge, I think you will know why I'm talking about all these things. Also, have you planned to integrate the immunotherapy approach in your research? If you are like I've given you example where my work actually took us to immunotherapy. It's not we wanted to do the immunotherapy. During that time that we didn't know what's going on, then immunotherapy came, and then our survival was better. So like that, there is your successes and challenges. There is always comes in between. There is success, there is a challenge. Okay, you are successful, and then you be happy, and then the challenge comes. So it's very common in research. So when it comes to combination strategies, overcoming a compensatory mechanism. So it's very important. When you target one, there is another signaling pathway will get activated. It's because the cancer cells want to survive. That's why the other signaling pathways get activated, and they want to survive. It's very important, very common. So that's why you need to understand the multi-signaling, which pathway is getting activated. Also understand the heterogeneity, like I was showing you. I tried to target the tumor intrinsic pathways, and then we did not get the survival. Then I went into heterogeneity study, and then we started inhibiting heterogeneity, whatever that immunotherapy, and then try to do the multiple approach to hit the cancer. So combining targeted therapy with immunotherapy, I gave you examples, things like that. But important is that how do you design your combination strategies, why you want to design combination therapy. Sometimes your data takes you there, sometimes you design and then see what happens, things like that. And also you need to think about biomarker study, that we did not touch much about it in the patient care, and biomarker study after you do the treatment, and to stratify the patient. Basically you do the biomarker study after you do the therapeutic studies. You collect all these blood samples. There are different ways. There are many ways that you can do biomarker studies. Translation research very important when it comes to a cancer research. That is the bench to bedside we always call. So and then sometimes it is important to also take from the bedside to bedside. Means that the patient samples, patient blood samples, patient data you collect, and come back to the lab and analyze them and see what's going on, and then you start thinking about the science. So that you can actually say bench to bedside, bedside to bedside. So it's important. Both sides are important. So think both sides. If you have opportunity to work with a clinician or the doctor or someone, it's actually great, because you're actually talking about the bedside to bedside. You as a researcher, you bring those samples back to the bedside, and then start thinking about your research as a clinical translational. So integrating the preclinical model to therapeutic strategies. I talked about today morning, like why you want to do the in vivo studies, because you want to take to the clinic. That's the reason that you want to do the clinic in vivo studies. The same way you have to understand the preclinical models, and also strategy, therapeutic strategy to. So that will tell you what's going to happen in the clinical trial. Maybe it will work, maybe it will not, but at least the logics scientific logics has to be perfect. So the importance of biomarker development, patient selection, and adaptive trial design. These are all going forward after phase 1 trial, and further when you go to the higher up, phase 2, phase 3. These are all comes in. Don't worry right now, but I think important to know all those phases. And there are challenges when translating your laboratory findings to the clinical setting. There is no doubt about it. Okay, there are a lot of learnings, and there are a lot of interaction you have to do with the clinicians, the doctors. So don't leave anyone. So any hospital, I was just keep telling, there are many hospitals here. There are many clinicians. They are knowledgeable, much more than what we think. Okay, so we are just a biologist. Okay, the information out there, what they are seeing the patient every day, and they have a huge information, real information. Okay, but what you have is the brain of thinking about biology. So you need to collaborate with someone like that, and then challenge your research, what current research you are doing, and challenge yourself every time. That's most pressing, important thing. How do you envision the overcoming the gap between the preclinical models and the clinical outcome? You have to start thinking yourself that these models, if you want to model this also, you need to have the collaboration with the clinicians or someone who actually knows. For example, I wanted to design the chemical library, and I went and had the discussion with the INSTEM, my friend. So INSTEM, he was able to help me. So because like that, we there are many of them. Okay, like different field. Pharmacology, there is another person called Raj. He's in Minnesota in another university, and whenever I have an issue with the pharmacology, I talk to them. So it's important to know pharmacological side, and how to develop the drug, and the problems and issues. And same way in food technology, you talk about the packaging I was talking about yesterday, and also there are a lot of other things that issues comes, and I know those process also. As when I was in CFTR, I saw that the whole in the spice and flavor technology department. So what I'm saying, each component, each when you want to design, there are always problems. You have to overcome those gaps and problems. In also cancer research, there is a from preclinical to the clinical outcome. There is always a gap in between problems. So we think about all those things, and when you are designing your experiments. So again, our last take-home message is there is no single model. Therapy is universally effective. Okay, so do you think that this model is perfect? There is no way. So there is another model comes out, and other model may be much better than you think. There is nothing called like single model is important. This is effective. If someone talk about one model, this is a great model, blah blah. It's okay, fine. You accept it and you move on and you do your best. Okay, this is what we did. Okay, we did whatever we think that is best. Okay, integrated approaches tailored to the specific cancer type and patient profile are essential, because you have to integrate approaches. Like means that in each cancer specific there is an approach you have to give. Okay, in my cancer I understand how I have to do. Okay, the same way if you ask me prostate cancer or some other cancer, so I just don't know how to do it because I'm not the experienced investigator. So but I only give you a hint, whatever I know from my cancer, give you a hint. But the probably the specific cancer type, understanding it, and also the specific cancer patient profile, understanding that is, I think, a key factor, essential. Okay, whatever I said and talked, okay, whether you're using it or not, but you will use it in the future. You have to start thinking today, so that it takes years to establish those concept ideas. It is not today. I know that you will not be able to today, but do it today. But the ideas and the concept and designs, how we think. How we think about the problems, and what and all you need to think about when you take one problem, right? One cancer, the same way, one problem, your scientific problem, it's those logics, I think, key thing overall, right? That's all. So we are done almost. Okay. Thank you so much. Now, a few minutes, we're going to have interaction, few questions, final questions. After that, we'll disperse, and if anything, we can discuss later. Okay. anybody have any general questions or anything about, anything, it's fine. Okay? Yeah.
Sir, it's not about science now. I think you told everything.
Yeah, it's fine.
Tell us about your journey from a small village like Nagatihalli to the US now. How is it? How was that? And what are the guidelines you want to give to someone who is aiming that?
So I'm a village boy. Okay, I studied up to I think seventh grade in my village school. My school has 100 years of celebration next week. Also, I'm doing that. I'm going to my village and we'll be celebrating for 3 day or 4 day function in our village, and there'll be a lot of a lot of students come, almost everyone probably trying to be there, and that is a huge thing. And after seventh grade, I think I was not a sport boy. I was always kind of when I now if I look back and we had. It's like a village, anybody from village is the same thing I have experienced, right? I have to take care of the home, and we had a goat and bullocks and all those things. Same thing, we had to take care of them. Saturday, Sunday, we had to take them to a coconut trees around, and we had to clean them. We had to put water to coconut trees. It's not easy, and also we had to take all the clean everything, all this goat and everything. We had to take those to the trees. We had to walk all the way to 2 kilometer, 3 kilometer, and put them back and come back and have a breakfast and go to school. And then afternoon, same thing. It's the same story, right? And we do all those things. And then I went to high school. In the high school, I think I went to Navodaya in Chandrapatna. So I was supposed to be in the English medium. They put me. There is an entrance exam for that, and I was supposed to be in English medium. So according to my dad was a teacher and headmaster, so he put me in, at least you should study. So, but I went into English medium, and then I realized that I was not able to, get anything what they are talking about. What I studied, social studies, is different than what they are talking about. I have no idea what those words are. Then I passed and I went to English medium, and then it came back. And I went to headmaster, and his name is Putaya. I still remember I did a namaskar, and then I told him that I will not be able to really understand any of those words, and I want to go back to Kannada medium and study. And then he said, okay, you know what? He was so happy that someone is coming and wanted to go to Kannada medium, because everybody wants to study in English medium during that time. And then I went back to Kannada medium, and he was so happy, and then put me in one section. And then I studied 3 years. My parents didn't know that I studied in Kannada medium until I got the 10th marks card. And I was a union leader, and I was a president of the union during that time. So my dad thought, I think he's going to go, I think become something politician or something. So then he put me in Mysore, some hostel. And then then he was really worried that this guy is going to go in other direction. So there was one thing that I disciplined myself, because I joined NCC during my high school. I was really a kind of a social boy, and NCC actually helped me a lot. I came to Mangalore during that time integration camp when I was in, I think, 9th grade. So in the Canara school here, I think I had a national integration camp. I think that changed my mind. That means that, when they call me Mangalore University, now you know why I'm here, because that inspiration, that when I was a small kid, when I had that integration camp from Punjab, from Assam, there are many students came from all over. I really saw all these people, different, language, Hindi and many things. That was actually, I never went out anywhere other than like Chandrapatna and then Bellur, and that's all. That's it. That's my whole, understanding of the whole world. And that I think integration camp helped us. So you all hear from Kerala and other places, and I think that is actually helping you, because you're seeing lot of things that you learn, right? So young age, if you get to know all this integration, of different culture and things, I think that changes your mind, right? Probably my mind is also was changing. I was actually leadership little bit qualities that during that time. Also, I was doing the Hindi Prathama, Madhyama, Pravesh. I was learning English, Hindi during that time, so that helps me. I was always kind of because I'm in NCC, I was maybe thinking I want to join army if I don't get anything, any job. Kind of that culture, that the high school is like, blood, how that flowing, like, you want to, do everything. Then when I went to Mysore, right, that is one of the, I never left Mysore until I did my PhD. Even though I got a lot of opportunities outside, when I, IRA and other things, but I came back always Mysore because very attractive education-wise. I think that place for me at that time, there were there were a lot of things that I learned. I used to attend a lot of lectures from different places, and also different colleges I used to go if their lectures or anybody is giving a talk or anything. It's my own discovery. You know what I'm saying? So when you're in a hostel, you have a bicycle and then you just keep going anywhere you want, right? This hostel was like only 2 meal, morning and then evening. That's all. There is no in between any meal. My dad used to give me $225, 225 rupees. Then 25 rupees extra, that is for the cycle, bicycle, 250 rupees actually every month during my PUC. PUC, my English medium. So basically, I studied, I told you, I think with a dictionary, I was always keeping my dictionary right next to my physics book and these things. I never went to any coaching classes or English, any mathematics or science. Even though I tried to go, but there was no money. You know what I'm saying? So all my sister and brother, everyone was studying. My teacher kids, and I think it's very difficult for everyone. Everyone was studying. My sister was studying in Mysore, and my brother was studying in Bangalore. Things like that. It's very difficult for a teacher to afford everyone. So we knew all those things. So kind of it is difficult for the tuition. So I learned myself everything because I think that hunger, I think, makes you better, right? It's important. Even though we say that when you are giving everything to our kids, what happens is probably without hungry, I think it is very difficult for them to understand the value of the food. So after the PU, I still went to NCC because that is my passion. Probably at least I will have a job in army, something like that. If I don't get anything. So went to after PU, I did not get that great score, so I had to go to BSc during that time. So at least I thought after BSc I will do the medical. I was very much interested in medical school. I was so passionate about medical school. And I didn't know how to do the CET exam. Believe me, I didn't know the training. Because I didn't have money to go to CET classes, course, or anything like that. So it was difficult. So we didn't get the score. So I was, I was not the great student probably at that time. But I was focused. Whatever I learn, I always get it inside. That's what I think. I attended classes, I think, regularly. I did not miss classes. So any classes, whatever I can get, 1 minute, anything, the knowledge I get from the classroom, I think that actually helped me a lot. One of the English teacher, I think I studied in JSS College in Mysore, PU, and then went to Yurajas College in BSc. In the PU, I think one of the English teacher, I think, attracted me a lot. He was actually teaching me English in Kannada style. Kannada to English, English to Kannada. So that actually helped me a lot. I had to really salute to him because that was needed for me at that time. Because that I was actually transitioning from my own language to English. So that's, I think, the key important thing. And there was a mathematics teacher, one of the lady that actually attracted or the way of teaching and all those things. So those kind of small things helps you all. I think you have the same type of experience for sure. You have a favorite teachers. And during my BSc, I think one of the research that I want to take today because of that my teachers during my BSc class, BSc time. There was a Panduranga, his name is Dr. Panduranga, Dr. Munyappa. He was a zoology teacher, okay. So Dr. Panduranga used to teach by taking the science and nature published papers on the front page, okay. When he was teaching me nurture and nature, he used to talk about, okay, how you kindle in the classroom. He used to take about nature, nurture, and say that this is not from genetics, this is from not from your parents, this is actually you. You're actually funny because your genes are just not from, this is not followed from, this is not a nature, this is nurture, kind of, something like that. And also, what you were saying, the same thing that has been published here. Look at this, nature and nurture. Exactly what you guys are doing here. This is exactly what it is. This is from the genetics from your parents, this. And this one, this is this, not from your parents. This is you, because you're going and talking to this in the park and sitting there, because this is not your parents. This is you, don't want to study, you have other friends, and you just have to, you have other girlfriend or someone is sitting in the, bench and talking nonsense and all things like that. So he was giving us a real picture of how to study. Means that take the one that is going around you, what you are, and think about it. It's actually that is what is the science. So science is about the biology attracts everyone. You're all biologists, kind of, because it's actually happening, right? It's not physical phenomena. It's happening, and that's why you're studying. You're attracted to it. That's what I think motivated us. That he taught us by taking the research articles, not by textbook, not by anything. That research is actually changed a lot of kids, not just me. Many of them in our college days. So I think as a teacher, it's very important how much you learned and how much you are learning, how you are actually teaching to a next generation. If you are not passionate to teach, you should not take up the teaching. Okay, you had to have the passion to teach. So those teachers are passionate completely. I mean, that is overwhelming. Actually, not just me. Everyone who actually studied there, zoology. And like that, there are many other teachers in many other classrooms, right? There was a Padmaji, he is a chemistry teacher. He was actually always with a suit every day. It doesn't matter summer or the winter, because from there we learn the discipline. Because he went outside the world, country, and then learned and came back. He was always disciplined, perfect, and you don't want anyone to be he's pin of silence, everything. So is this discipline you learn from him, there's so much. Like his name is Dr. Padmaji. So like that, you all learn from each teacher something. So I learned those things probably. And when I went to, when I wanted to become a doctor, I think I wanted to do the medicine. But believe me, during that time, that year, the entrance exam came in Mysore University. That was the first entrance exam. There was a Kaveri Galata, and then everything was happening, and then entrance exam was postponed and then postponed. We had long holiday, and then finally entrance exam happened. That's the first time entrance exam, because there was a fighting. There was not to have the entrance exam for masters. So the entrance exam, then I cleared. I was cleared MBA. I was in the botany and the zoology, and then when I got into a botany, this is a story. You asked me story. I think important, because you all have a story to tell for sure. And this is my story, and probably you will have your own story. Probably you will be even fantastic. Yours will be probably when you become someone. I think probably start talking about all your background and everything. So when I got into botany, and I wanted to go to zoology, because I had a professor, Dr. Rangnath, who is a NAC director, and I was related to him. So I was former NAC director. So I wanted to really do zoology, because the zoology attracted to me during my undergrad. And somehow I did not get it in the first list. And so I got in the botany, and then I joined botany. And then I was not disappointed a little bit, because zoology was attracted, right? And then during that time, I was in NCC, and then I had a NCC during that time. Even my BSc or NCC, I was a C certificate, I was an under officer, and also I went to a lot of integrative camps, and then military camps, and BLC, and a lot of things. I did a lot. So that's why I did not study so well. So I was doing a lot of other integrative. I went to a lot of places, military attachment camps and leadership camps and many things. So during that time, I did get a lot of scores. Then went into NCC. I scored a good number, and then through NCC quota, I got into biochemistry. So, because biochemistry in Mysore University during that time, 20 seats, right? There were no other biochemistry in the all over Mysore University. There was only one department. And in biochemistry, I think 20, most of 15 people will be rank students, all of them rank students. And then we have 5 from the other categories and sports quota and other things, foreign quota and things like that. But it was tough for general biochemistry students to do the biochemistry. It was not easy. My own friends were there right next to me. But, it's like tough because all they are from the BBM, biochemistry, microbiology, but I'm from the chemistry. So it was challenging. So we did study though, but it was very challenging for the general chemistry students to do the chemistry, biochemistry. But we had great teachers. Okay, we had great teachers, and T.V. Veerabhasapagowda, and then MKK is, and then many teachers. There were a lot of Cletus D'Souza. I think he's from Mangalore University. He was also my teacher, and many more. Okay, the great teachers I think changed our way of thinking science, right? Way of thinking the research. The Veerabhasapagowda, TVG we call. He was a professor, and he guided me during my master's thesis. And I think there are a lot of things we learned. A lot of things we learned. From chemistry, I think teachers were attractive because Rangappa and Timaya during that time. There are many other chemistry teachers after that, and zoology. Many Ranganath is one of the geneticists. I think. I listened to him. He gave me one book, genetic book to read and study. During that time, I still remember. Like that, many teachers were there, just not them. The great campus, attractive campus. But important thing is that I think key factor when I was in bachelor degree. I know Joshi knows. I think I used to go to Mansa Gangotri, that is a campus, PG campus. I used to study in that library. So it means that somehow I had a librarian attached. Knew these. Through someone I used to hide and go and study because I wanted some peace area, environment. We were in the small rooms and very difficult. There were 3-4 people in one room and things like that. And during that time, cooking and all those things. So important, I think, if you want to really study, you will find a way. That's all I can tell you. So I was also Kannada literary. I used to read a lot of personalities in Kannada language. So I used to go to Kannada area, where if you go to Mansa Gangotri, there is a left side. If you go, there's a Kannada and section, and then history and all those. In the right, if you go to science, and there's a I think Joshi might have studied there also during his exam time and all those things. So. That's how the life is. After I think masters, you don't know what you want to do. Believe me, you want to become probably we all wanted to become a medical rep or things like that, because it was tough even during that time to get a job, and then we all end up in CFTRI become research assistants. That's what we did. Everyone. Become a research assistant because there are a lot of projects there, and that's what I did too. And but I felt that I'm wasting a lot of time, because by the time you have a master, and your parents are expecting something from you, right? And you have to get settled or get job, right? But I was not doing that. That is like 1,500 rupees is not enough. 1,200 rupees or something. It was not enough after masters, right? 1,000 rupees is like, just give a room rent, that's all. And so that actually, I went back and told my dad that I wanted to study more. I just cannot do this, because without studying further, I don't think I will be able to do anything, okay? So my dad said, even at that time, even the difficult time, he said, who asked you to go and work? I did not tell you to go and work, but go study whatever you want to do. Then I started studying for competitive exams. Did a lot of, there was a wing commander Raghunath. He was a great teacher for me. He was a army guy and who actually fought in Germany, and he was a Mysoreans know him very well. And even to go to his classroom, he's a private classroom, free classroom for a competitive exam. He used to teach history. I never learned history when I was in school time, but I learned there after my masters, history, geography, understanding the whole concept of historians and their lifestyle, and how important it is, and also personalities. I think development. Someone like him, I think a big difference in your life, right? There was a Narayan Swami is a G. Narayan Swami is well-known commissioner. Now I think he's in placed in somewhere else as he's a commissioner at that time in the income tax, and he was a botany MSc and he did IRS, and he was actually guiding us for essay writing, also competitive exam, anything like AS coaching. Something like coaching means that talking about, it's not that you can, they see, one thing I keep telling, even science, I cannot change you. You have to change yourself. Same thing. Narayan Swami used to tell me that you have to change yourself. Only he can actually guide you. Okay? He's such a great personality. When we go to his office, even though we were like masters kids, he used to give us, first thing he used to do is order tea for everyone, all the students, and after the tea, he used to discuss what you have written essay writing. For example, he will give you one of the writing, what you have to write, and then we write, and then we go there. That's how our writings are improved. Even though I do scientific writing, something that it actually always, you think about it, what the changes that you learn during that time, right? That it is for life. You in life, you just keep coming back, right? So this is a life lesson. Once I started studying, I think I started clearing UG's, any exams I take, any entrance exam, any competitive exam, because you get that confidence, right? Some kids, I think it's very difficult to get that confidence. Some kids, they do it just like that, right? So for some kids, like me, probably I needed that whole environment after my masters. So that changed the way I think about competitive exams or any exams, are going to the interview. So after that interview, I cleared and went into DFRL, because I wanted to go to being local, because I was studying for the competitive exams. So and it helped us a lot. But as you enter the scientific world, when the competitive exam did not go through the way we think, right? Because as an background, what our background is, and also having a family with 4 kids and things like that, and having a fellowship, DRDO fellowship, even 2,500 rupees is a great thing at that time. I had a lot of money. I didn't even buy a bike, in a loan. What I'm saying is that 2,500 is a lot. And that was actually changing, right? From 1,000 rupees to 2,500 is a lot of money. And then the PhD, I think having a right mentor. My mentor was a IASC PhD. I think he was a DFRL director at that time. He was a department chair biochemistry, and then that actually mentorship, I think a little bit. Dr. Santanam, he was a great mentor because of the experience. He went to US and then had a postdoc and came back. So yeah, he knew the world, he knew what science is, and then he is from well-trained institutions. So the mentorship, I think you have to have a mentor who actually have been trained well. You have to find a mentor who actually went outside and world and has seen a little bit here and there. Having a right mentor changes you a lot, changes your life too. So at least whether in mentoring or not, the personality is a key thing. So what type of personality the mentor has is a key factor for your life to change. Because whether they're going to teach you or not, just having a personality is also important. So that type of personality, even today, I just messaged yesterday to my mentor. He's in Bangalore, so I'm going to meet him next few days. But important, whenever I come, even when I was giving any lecture anywhere, he comes there and he is so happy to be there. You know what I'm saying? So it's about that type of mentor is a key factor. Like I'm coming from somewhere and giving a lecture. When he comes to Boston, his kids are there. When I go to Boston, I met him one time, and he was so happy. So it's important, that relationship that important for your life. Whatever the mentor is, that personality is important. keep that in mind, that it doesn't matter your stage now or later. I have a mentor through all my career. Even now, we have a mentor too. Mentor means that someone is guiding you through something. There's always, you have to find mentor every stage of your life, is a key factor, right? It's not that you're not over. Even though you're a professor, you have to have a mentor who actually guide you to the next level. You can actually guide you what else you can do. It's not over yet. You know what I'm saying? So those kind of relationship and important. After that, I think I gave you already hint at how I did my research and what difficulties were there. And even though there are many difficulties, that I was at DFRL, we had a lot of problems too during that, because it's a defense laboratory. They didn't know the research, JRF. What is the JRF means? Means that in the defense environment, you guys may not think, because I know what it is in a defense environment, because during that time it was like that, but now it has been changed. And believe me, there are a lot of researchers coming out from those institutions, and DRDO fellowship is a great fellowship. Like any other fellowship, any CSIR fellowship, it's a competitive. So it's that institution is changing, okay? And you need to have the institution, university is also important, and for a base. And you don't have to learn anything from it. You have to, end of the day, it's you. You change and you want it, you will get it. If you don't want, you will never get it. Okay? And if you blaming anyone, again I'm repeating, so you'll never get it. Okay? You just have to change yourself and keep doing what is right for you and your life. Okay? So end of the day, you change your life, no one else will change yours. So that's what I think I learned, those tips on the way. So I really wanted to do research. That's the reason that I went to US, and I think there are a lot of tough times. Believe me, it is like it looks great from outside. It is not like that. Okay, there is a lot of tough life out there too. Okay, there is a lot of crying. There are a lot of struggle. There are a lot of you go through, and there is the food issues, and you have to cook. Otherwise, you will not get a food. And you have to cook yourself, and you cannot eat burger every day. And you want to go to burger shop, and there is a culture shock. And we learned all those things, right? And many days there was even we didn't know whether there is a hot water and cold water in the shower at that time. You know what I'm saying? I was always taking cold water. I didn't know there was a hot water also other side. Things like that. But my aim was to do cancer research. I would do research, something to do. But my intentions were not anything else. But I knew that I can go anywhere, and I can survive and do. You know what I'm saying? I took like $100. That's all when I went to US. That's all I had in my money in my pocket. My mentor gave me a check. She was like, really, you have like this much money and you want to survive in US? I was like, she was shocked. And then and she gave me a check, and then I had to write a signature saying that after my first salary, I'll return the money. So that's how I think Eugenia Wang, her name. So she gave me that first check, and that's how we have survived. So okay, if you don't have money, also you can actually survive. You don't need. There are people, but you have to believe in yourself that you can do it. If I were to say, like, I don't have money, I cannot go to US, somehow, you know what I'm saying? So I went, and so we survived. I didn't know anything about anything in the world, okay? I didn't know how to, first of all, I didn't know there is a hot water, right? Think about it. I didn't know anything, okay? So, but we learned. But my focus was this, research. That's why I went. There's nothing else, right? So, on the go, you learn everything, either, cooking to anything, living, and having friends, and having a friends like, someone from institution like IASC is a great thing, right? You get everything there because it's hard to go get it here. When you go outside, they all become friends, and you learn from them, and you share yourself, and they share. Initially there was a struggle. There were other people who were publishing great papers. I was not. You know what I'm saying? But what do I do to get those type of paper? I left the university, went to Vanderbilt. It's like a Harvard University when I got a chance. Because to do better science, to do more models, do more in vivo, translational, things like that. You have to challenge yourself. There is a lot of other jobs come in between. I would have become an assistant professor. I would have become something else in there also. I could have gone to food nutrition biochemistry. I would have become assistant professor. That was not my intention. The intention was to do cancer biology. So I had to survive, strive, and go to other universities, learn, and struggle. And again, you know what I'm saying? There are a lot of family struggle, life struggle, and then a lot of things. There's story, a lot. 25 years of story. You want to hear a lot. Okay, there is struggle everywhere, every day, and end of the day, I think you all will be happy. So we are all happy. Doesn't matter how much the struggle we go through, right? So you all will be happy. I think you all will see the happy ending, right? It's important. You all see it. I think you should not forget one thing is that who train you, who guide you, and like I was saying, like my mentor, I always see him and try to talk to him. It's important because even I become mentor too. I do the same thing to my next generation. It's the same thing. This is important when someone is teaching you, and you should not forget. And also someone your students has to be like that. So you have to train them such a way that humanity exists and will be there forever, right? So that's the story. Thank you.